from the perspective of 3D structures. So Dr. Yue gave a good introduction to high-C data. So the high-C data is typically represented in the form of contact matrices. However, these can be embedded using dimensionality reduction techniques such as multidimensional scaling. They can be embedded in 3 dimensions to give structures representing the ensemble average of the population of 3D chromosomal conformations. So, for example, this is showing high-C structures from GM12878 chromosome 21 and K562 chromosome 21. So GM12878 is a lymphoblastoid cell type, K562 is a cancerous cell type. So once you have these 2 structures, the question is how to compare them. And it turns out that this is pretty difficult, partly due to the stochastic nature of MDS. So I've developed an alternative approach called MultiMDS that takes 2 high-C data sets as input and simultaneously infers structures for both of them and aligns the structures. So how the algorithm works, it's a modification of MDS that involves first holding one structure constant and finding an embedding for the other structure that simultaneously minimizes the difference between the input data and the embedding and the difference between structure 1 and structure 2. And then you iterate between those 2 steps until convergence is achieved. So once we have these aligned structures, it becomes straightforward to compare them. And this gives us a locus-specific comparison of the high-C data, because for each locus on chromosome 21, say 100 kb bin, we can simply calculate the Euclidean distance between the structures, and so a higher distance would indicate a greater degree of relocalization in the nucleus between these cell types. So to validate my method, I wanted to use a fairly orthogonal way of looking at high-C data, which doesn't involve any sort of structural inference. So you may be familiar with A-B compartmentalization, which is based on a principal components analysis of the high-C contact matrix. The A compartment has shown to be active and the B compartment has shown to be repressed based on markers such as histone modifications. And it also turns out that these compartments form a single axis in the nucleus with more higher B compartment scores corresponding to the nuclear periphery and the A compartment corresponding to the nuclear interior. So this is significant because it means that all compartment changes correspond to relocalizations within the nucleus. So this is an independent way of validating my method. So I find that across all chromosomes for the GM12878 K562 comparison, the MultiMDS method shown in red has a higher correlation between the measured relocalization and compartment changes compared to other methods. So there aren't really dedicated other methods for doing locus-specific comparisons of high-C data. So I just used some simple methods for comparison. The Pearson and Spearman correlations are ways of comparing vectors. So it's been previously pointed out that these are not very suitable for high-C data, and I find that here as well with them failing to capture compartment changes. So then I wanted to quantify how much of the relocalization within the nucleus between different cell types is occurring along this compartment axis. And I find that for most chromosomes, there is an enrichment of changes occurring along this axis. So what I'm measuring here is the average distance moved along each of these axes between the two cell types. However, compartment changes have been pretty well explored already, so I wanted to explore changes that are occurring independent of compartment changes. So here I'm showing a single chromosome, chromosome 22, and along the x-axis is genomic coordinates, and in blue I'm showing the degree of relocalization at each locus, and in green I'm showing the absolute compartment score change. So I did peak calling for each of these signals, and you'll see that many of the relocalizations correspond to compartment changes, as I showed before, but I wanted to focus on the relocalizations that were occurring independent of compartment changes. And then further, I focused on changes that occur within the A compartment because that's the active compartment. And I find that these changes are enriched for TAD boundaries that change between the cell types. So though TADs are conserved overall, and this is still a small fraction of the relocalizations, compared to the shuffled TAD boundaries, I see a significant enrichment of TAD boundaries that overlap with my relocalized loci. I also find that these intra-A compartment relocalized loci are enriched for very broad regions of H3K27 acetylation that some people might refer to as putative enhancers. And this is even compared to the A compartment itself, which tends to be enriched for active marks such as this overall. And looking at these intra-A relocalized loci that only have one of these H3K27 acetylation peaks in one cell type, so there's a cell type specific loss of K27 acetylation, I wanted to see what was happening to those loci that lose that active mark. And I found that there's significant differential looping at those sites, and the differential looping partners tend to, while they tend to be in the A compartment, they gain enrichment for H3K27 trimethylation repressive marks. So these loci seem to be moving into more K27, more repressive regions within the A compartment. So in summary, I find that while the majority of relocalization between cell types occurs along the AB compartment axis, there is a biologically significant variation that occurs orthogonal to this axis. So even within the A compartment, there's significant reorganization that's enriched for differential TAD boundaries and also for enhancers, putative enhancers, represented by long-range H3K27 acetylation. And the loss of these K27 acetylation peaks is correlated with a movement into K27 trimethylation enriched regions. So my method is publicly available. It's called MultiMDS. It's available on GitHub. And I think with the availability of high-C data sets, there's really there's a lot of opportunities to compare, for example, time course differentiation, and also comparing condition-specific high-C data. So for example, with a knockout, how is that changing the high-C data overall? So with that, I'd like to thank my PI, Dr. Sean Mahoney, who will be presenting later in the week, the rest of my lab, our close collaborator, Dr. Frank Pugh, and his lab, and my funding source, NSF. And I'll take any questions now. Thank you.
Do we have time for maybe one question?
Mm-hmm.
If there is any. So I'll quickly ask.
Mm-hmm.
So for your comparisons, we are looking at mainly GM12878
Mm-hmm.
and K562.
Yeah.
K562, as you know, is a cancer cell line,
Mm-hmm.
right? So some of these changes, you cannot deconvolute whether they are because of the copy number differences, CNVs. Why would you not take a regular, normal, diploid cell line instead?
Well, I was interested in K562 because in some ways it's closely related. It's a leukemia cell line, so it's more closely related to a lymphoblastoid cell line. Also, there's quite high resolution data available, and the strength of this method and structural inference in general really comes from high resolution. I did take care to eliminate any known translocated regions. I eliminated them from both cell types. Thanks.
All right. Well, thanks, speaker. Our next and last Penn State talk is going to be from Ta Young, and then he's going to talk us about the differential